Hello, everyone, and welcome to today's webinar, A Novel Strategy Targeting Myeloid Cells and T Cells in Renal Cell Carcinoma. I'm Melanie Chan of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit www.thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technicalities, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speaker, Maurice Alcantara, postdoctoral fellow at the City of Hope National Medical Center. For a complete biography on our speaker, please visit the biography tab at the top of your screen. Maurice, you may now begin your presentation.
Hi everyone, before I begin, I just want to turn off the camera, but let you know I'm going to be here just for the purpose of focusing on my talk. So I'll be off camera, but I'm still right here. Thank you, Melanie. So everyone, thank you so much for making the time to attend my webinar today. And as mentioned by Melanie, my name is Maurice Alcantara, and I'm a postdoctoral fellow at City of Hope National Medical Center in Los Angeles in California. I'm so honored to be part of the Gibco Cell Culture Hero webinar series, and I really appreciate the chance to discuss a little about my work. I'm also thankful to both Thermo Fisher and Gibco for the chance to talk about my work, given both companies have been integral to my experiments. I've actually used them quite a lot in some of the experiments I'm discussing today. So as discussed, I will be talking about a novel strategy targeting myeloid cells and T cells in renal cell carcinoma. Just a brief overview, I will be talking about my research career, renal cell carcinoma, the current therapies in renal cell carcinoma, and the challenges that are still evident in the disease. STAT3, the transcription factor that I was targeting, and the current strategies in STAT3 targeting and the challenges associated with them. Before going into T cells and myeloid cells, our lab's novel strategy to target STAT3, which is CpG STAT3 antisense, and then some results, a summary, and last but not least, acknowledgement of all the people who contributed and assisted in my work. My research career really began after my undergraduate degree when I did an intensive year, or what's referred to as an honours degree in Australia, where typically students do a very intensive 12 months of research and then write a thesis at the end of the 12 months. During this time, I developed a deep appreciation for science. And I was really mentored by a very kind molecular biologist who really helped me ignite my passion for science. But what really captivated me most about science and the scientific process and being a scientist is the ability to take ideas that are kind of abstract, that not many people understand, and it's all in our head. And then through design and experimentation, prove or disprove some of our ideas that's only in our head. This process continues to inspire me today. I then went to do a PhD in Australia, co-mentored by an immunologist and a physician scientist. I did it at quite a small institute, but this is when I really developed a passion for immunology and a deep appreciation for the role of our immune system and our immune cells in both health and disease. I gained really important experience in working with human samples and research, and my favorite technique, which is flow cytometry. During my PhD, I was running and maintaining our own flow cytometers. It was the BD Fax ARIA 2 cell salter, which is now quite old, but at the time was cutting edge. And it was quite rare for a PhD student to have access to their own. Typically, people had to book in core facilities, so I felt really lucky. So this experience in my PhD and the knowledge that I gained during this time was able to get me a position at City of Hope, which is located in Duarte, in just outside of Los Angeles. And for the past 6 years, and continues to be my home in research. As of physicians, physician scientists, translational scientists, and basic scientists. Because it's home to both patients, doctors, and scientists, it's really a great place to do cutting edge research. There's research here ranging from preclinical testing of drugs in cells and animals to phase 1 clinical trials in patients. Because we have access to cells, to animals, and to actual patients and their tissues and samples on site, we have the ability to do great translational research. The main focus of my research for the past 6 years is renal cell carcinoma. RCC is the most common type of kidney cancer. It actually accounts for 90% of the disease. It originates in the nerve tubules, which are responsible for filtering waste and balancing body fluids. The exact cause of RCC remains unclear. However, there are several risk factors that have been identified, including but not limited to a genetic predisposition, smoking, and chronic kidney disease. The disease encompasses over 10 histological and molecular subtypes, of which clear cell RCC is the most common and accounts for the most cancer-related deaths. What's interesting is that RCC metastatic disease represents about 30% of this disease and is refractory to all current strategies. However, most patients will ultimately develop metastatic RCC. The current strategies that's utilized to treat renal cell carcinoma. So for localized disease, we can successfully manage with surgery, ablation, and active surveillance. The problem really arises when you have metastatic RCC. And about 30% of patients will develop metastatic RCC. As I said, it's refractory to conventional chemotherapy options and really requires systemic therapy that typically targets VEGF, mTOR, and recently immune checkpoint blockade has been approved in the disease. And as you can see on the right, immune checkpoint blockade has slightly improved survival in patients. However, there's still a need to improve patient survival because it's only slightly improved, and the 5-year overall survival rate for this disease still remains at approximately 30 to 40%. So this suggests that there's still a lot of room for improvement and a need for the identification and development of new therapeutic strategies and new targets. So this brings me to the immunology of renal cell carcinoma, which from my perspective is important to understand not only why patients don't respond or become refractory to treatment, but also holds the key to finding new targets and therefore new therapeutic strategies. Clear cell RCC, which is the main cancer that I focus on and most of my patient analysis is focused on, is unique in comparison to other tumors. It is responsive to immunotherapy. It has a low somatic mutational burden, and this is not associated to immunotherapy response. It has an abundance of CD8 positive T cells, but it's not actually associated with improved immunotherapeutic response. Tumors have a heterogeneous microenvironment that consists of many different cell populations consisting of myeloid cells, tumor-associated macrophages, as well as DCs and T cells. So from my perspective, new strategies need to consider targeting all of the immune cell compartments in the tumor and not just the way that it's doing it currently, which is targeting primarily only the T cells in the microenvironment. And so this brings me to talk about transcription factor STAT3, or signal transducer and activator of transcription 3. It was discovered about 30 years ago, and it is a transcription factor that was implicated in cancer inflammation. It is constitutively activated in numerous cancers, including renal cell carcinoma, and it's been established as a master regulator of tumor evasion. STAT3 is always activated in many cancers and promotes the survival, proliferation, and proliferation of many cancers, ultimately leading to resistance to therapy. Briefly, STAT3 is activated typically by cytokines in the microenvironment, and it is phosphorylated and translocates to the nucleus, where it then regulates gene expression and ultimately influences immune function and then tumor growth. As a result, from my perspective and our lab's perspective, targeting STAT3 may be beneficial then to improve tumor growth, promote tumor apoptosis, promote the antigen presentation of all the other cells to then support T cell function in the tumor microenvironment, and inhibit tumor promoting inflammation in the microenvironment. So there are already numerous strategies available that target STAT3. However, most options are small molecule inhibitors, which target STAT3 non-specifically. Because STAT3 is not only implicated and plays a role in cancer, but it's also important in normal immune function. This global inhibition that you can achieve with small molecule inhibitors is an ideal because it can result in immunosuppression. That's not the best strategy. In addition, STAT3 is also difficult to target because it has what's known as a lack of druggability. The lack of active sites make targeting it with small molecule inhibitors difficult, so it's not efficient. Then there's also the feedback loop and redundancy. If you're targeting it globally and unspecifically, you might activate alternative survival pathways, leading ultimately to drug resistance. And for this reason, a targeted approach to STAT3 inhibition is ideal. So this brings me to my lab's design and strategy to target this. So my PI developed a strategy where he conjugated a STAT3 antisense oligonucleotide, which silences STAT3 expression to CpG that is only internalized and recognized by toll-like receptor 9 expressing cells. This means that it's only internalized by innate immune cells, leaving the T cells out of the picture. So typically these are your myeloid cells, your dendritic cells, your macrophages, but not the T cells, which are the primary anti-tumor effector cells and the main cell types that's already being targeted by most of the drugs that's used for this disease. Our team has shown previously that this strategy is effective in inhibiting tumor growth in various mouse tumor models. However, my main focus was seeing if this can improve tumor growth in renal cell carcinoma. So this brings me to talk about some of my results. I covered previously that RCC tumors comprise of different immune cells, like lymphocytes, like T cells and B cells, and you have your myeloid cells, such as macrophages, dendritic cells, MDSCs, neutrophils, B cells, and tumors. Remember that the first line of therapy in the disease typically include immune checkpoint blockade and TKIs that primarily target the T cell compartment. So in collaboration with my colleague, we first asked the question, what are the main immune cell types that are present in patient tumors and peripheral blood? So I collaborated with my colleague, as I said, and we began analyzing patient peripheral blood and tumor match samples, single-cell RNA-seq data, and we observed that RCC patients had a lot of T cells, had a lot of NK cells, B cells, monocytes, macrophages, and dendritic cells. And interestingly, we also observed that there was a lot of overlap between patient peripheral blood and tumor, as you can see here. So tumor and peripheral blood. Because most of the current strategies are very focused on targeting only these cell types. As you can see, this suggests there's so much room to target all the other immune cells and really focus on combination therapy that targets all of these immune cells that are present in both patient peripheral blood and tumors. So I then analyzed patients who received T-cell targeted therapies at City of Hope from two separate clinical trials. So I noticed an interesting pattern. Patients who didn't respond to therapy, I noted them as non-responders, had significantly higher cytokines, for example, IL-6 and IL-8, that are implicated in STAT3 activation. This suggested to me that STAT3 activation in non-responders to therapy may potentially be playing a role in resistance. Interestingly, unsupervised clustering in these same patients also revealed that there was STAT3 activation, as you can see in red here, between baseline and week 12 in non-responders, primarily evident in the myeloid cell and B cell populations. So from my perspective, this suggested that myeloid cells and B cells from patients who receive T cell therapy may benefit from STAT3 inhibition. Indeed, when I analyzed these patients further, there was a strong positive correlation, as you can see here, between the cytokines in these patients, IL-6 and IL-8, and the STAT3 activation in a specific subtype of myeloid cells, primarily the MMDSCs, in only non-responders to therapy. That wasn't evident in responders to therapy. One of the key questions for me especially was, could these myeloid cells have an influence on T cell function, given that these T cells are the primary effector cells to anti-tumor responses? So I took T cells and myeloid cells from patients and co-cultured them. And what I found was that if cultured and stimulated alone without the presence of myeloid cells, T cells were more than capable of proliferating, and which is indicative of their function. However, if it was in the presence of myeloid cells and you cultured T cells, the T cells had less capacity to proliferate or function, suggesting that the myeloid cells were capable of suppressing these T cells. Interestingly, if I took our lab's drug, the CpG STAT3 antisense, and I co-cultured CD8 positive T cells and MDSCs, I observed that the T cell function or the proliferation could be restored. So this suggests that myeloid cell STAT3 inhibition in this renal cell carcinoma may benefit the T cell function and indirectly T cell targeted therapies. So then I began testing this idea in preclinical models of the disease. I took our lab STAT3 inhibitor with the perspective that if I could bind it with anti-PD-1, which is already utilized in the clinic, it may improve tumor growth control if I used the myeloid T cell targeted approach in these animal models. So indeed, what I found was that when I tested anti-PD-1, the T cell targeted drug that's used in the clinic, with our lab CpG STAT3 antisense that primarily targets myeloid cells, I observed an improvement in the tumor growth control, as you can see in red. And as you can see here, in preclinical models of renal cell carcinoma and also in bladder cancer, as you can see in red, where I can achieve complete regression of the tumor growth. I then analyzed the tumors of these animals that were treated with this strategy. And analysis of the harvested tumors revealed that there were several populations of immune cells that were targeted, mainly macrophages. Were the immune cells that was altered by the combination strategy. So I saw a decrease from the M2 macrophages, you can see here, the population that's most associated with pro-tumor microenvironments, and an increase in the antigen-presenting cells known as the M1 by macrophages, but also an increase in antigen presentation by DCs in the tumors and also the tumor-draining lymph nodes, which suggests that an increase in antigen presentation and a reprogramming of these innate immune cells underpins the improvement in tumor growth control in the disease. Finally, I also observed that there was an increase in T cell activity in these tumors, as you can see in red, which is the main cell type responsible for the anti-tumor response, which is what I wanted to see. So just to summarize, RCC patient tumors as associated with myeloid cells, which play a role in immune checkpoint blockade resistance. However, the role of myeloid cell resistance in the disease is not well understood. I also observed that RCC patients have alterations in myeloid cells and STAT3 activity, suggesting that STAT3 activation, specifically in these myeloid cells, may improve the therapeutic strategies. The results suggest that combination therapy indeed improves anti-tumor responses, and this is mediated primarily through improved antigen presentation, which supports T cell function. Overall, this shows that targeting immune cells as a whole is beneficial in the disease. Finally, I would like to acknowledge everyone in my lab and in the clinic who contributed to the work. Without these people, I would not have been able to do any of this work, and I want to thank them for their support over the last 6 years. Thank you so much.
Thank you, Maurice, for your informative presentation. We will now begin the live Q&A portion of the webinar. We have lots of questions that already came in, but if you haven't already submitted any questions, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Great. Now let's get started. Our first question asks, what is the therapeutic potential of targeting STAT3 in myeloid cells in combination with immune checkpoint inhibitors?
Okay, that's a great question. And primarily, when you're doing a targeted approach and you're targeting both cell types, because the immune system doesn't function in isolation. And right now, in many cancers, including this disease, a lot of the options only target one cell type. But like in this case, the immune system doesn't function in isolation. T cells require antigen presentation and support from the myeloid cells, and then the myeloid cells in turn can't effectively kill tumors. They rely on T cells for that. So the best approach would be like a teamwork approach. I like to use the analogy of like in basketball, the Scottie Pippen and the Michael Jordan. Michael Jordan, a lot of people know that he's a superstar, right? But he can only effectively function within a team that's functioning well. And I like to think of the myeloid cells as the team and the T cells as the Michael Jordans. And so combination approach and that teamwork approach really elevates the anti-tumor immune response. And that would be the best approach going forward, because our previous approach in therapy has been very focused on just the superstar one-sided approach, which is just targeting T cells or the Michael Jordans. And really what we've seen is that it'll work in 30-40%, but then you get like the other percentage where it doesn't work.
Great. Thank you, Maurice. Our second question that came in asks, have any clinical trials specifically targeted STAT3 in the myeloid compartment in RCC? If so, what were the findings?
Only indirectly, like they would not specifically target it. They would indirectly target it by target something upstream or downstream. So they have already used different inhibitors, but not specific to just that population. And so they were targeting all these other different things. And so there was a lot of off-target effects, which just means like effects you don't want, essentially, and it didn't work very well.
Great. Okay. The third question that came in asks, how does inhibition of STAT3 signaling impact the efficacy of existing RCC therapies like tyrosine kinase inhibitors or immunotherapies?
So if we're doing a very specific approach, it should really what's the word is like add to it. So that's why I'm really focused on like a more targeted, because we don't want to be messing with all the other aspects of the immune response and the treatments that's currently being used. So it should really just add to it and contribute to the natural efficacy rather than overtaking it. So that's why the current like clinical trials or the other drugs that don't target it very specifically, I wouldn't use that. Because it would just hijack the entire system and override the current strategies that's already being used. So I guess that's my question. That's my answer.
Okay, great. Our next question asks, are there reliable markers to distinguish STAT3 dependent from STAT3 independent suppressive myeloid populations in RCC?
STAT3 dependent and STAT3 independent. There's no really, because in general, if you're getting immunosuppression, we can't really tell what pathways has been activated. We can only say, I can only say from the patients that I have analyzed that it was STAT3 dependent because I've tested it. So we can't really say it's specifically that pathway that caused why these patients didn't respond. We would have to, going forward, do a more personalized test so we can understand the pathways that's being activated in each patient. And I think cancer therapy, that will be the future of cancer therapy, but as of right now, we don't have a way of doing that.
Understood. And it looks like we have time for one last question here. And it asks, in your opinion, does STAT3 inhibition synergize better with checkpoint blockade or anti-angiogenic therapies in RCC?
I think you mean checkpoint blockade because the other therapy, they're sort of in the same pathway. And so then you're targeting the same pathway multiple times. I think it's best to target different pathways and then combine it rather than the same pathway by different ways.
Okay, great. Thank you again, Maurice Alcantara, for your time today and your important research. We would like to also thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. We do not have time for it today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.